Bismillahirrahmanirrahim. Assalamu alaikum students. Hope so you are all fine. This is your science class and we are doing chapter 1 from cells to organisms. So today we will discuss about multicellular organisms. So you know that unicellular organism mean all those organisms or living things which possess unicell or one cell. So here we have to mention multicellular or multicell. Multi mean more than one. It can be two, it can be many, millions, trillions or billions. So students, living things such as plants and animals are made up of many cells. They are called multicellular organisms. A large animal such as elephant is made up of many more cells than a small animal like a frog. So students, as the size of an animal increase or size of a living organism increase, the number of cells from which it has made will also increase. So here are some examples of multicellular organisms. These are frog, elephant, moss and flowers. So students, you also know that like other animals, human being is also a multicellular organism because they are made up of many cells. How big are the cells? To find out, we can observe different kind or different types of cells through microscope. Basically, microscope is a special instrument which is used to observe microorganisms and to observe different kind of specimens. So students, here you can see that this is a microscope and this is an ordinary microscope which we use in lab. This is also known as light microscope because it has a light source in it from through which we can observe the slides easily. So now I will tell you about different parts of microscope. This is ocular lens or eyepiece, and this is the point from where the eye looks into the microscope. This is arm. We use arm to hold microscope when we have to move microscope from one place to another. This is stage at which specimen slide is placed. This is coarse and fine adjustments. Basically, these are known as focus knobs and they are turned to get a sharp and clear image. This is base. Base should be held together with the arm when moving a microscope. So when we have to move microscope from one place to another, we will pick it up from arm and base. So then we will move it from one place to another. This is body tube. This is nose piece. These are objective lens because these objective lens will be used to magnify the specimen or object. These are stage clips. The function of stage clips is to hold the specimen slide still on the stage so that we can easily observe the slide by moving the stage. This is diaphragm and this is light source. So light source will be used to generate light so that we can easily observe specimen. So due to presence of this light source and we have to observe things under this light, so that's why this microscope is known as light microscope. So next thing which I am going to tell you is specimen. So specimen is that object which we observed under a microscope. So all the object which we observed under microscope is called specimen. It is placed on a microscope slide. Here you can see these are all microscopic slides. which is then placed on the microscope stage, and this is the stage where we have to put this light, and then we will adjust focus knobs to observe clear and sharp image. Another point which is discussed here is cell theory. So students, cell theory was proposed in 1839 by two scientists, Sheldon and Chevron. So this theory contains 3 points. First one is composition of living things, that all living things are composed or made up of one or more cells. Secondly, if we talk about origin of cells, then cells come from pre-existing cells. Cell will come from those cells which are already present. Third one is basic unit of life. Cells are the basic units of life. That if there is no cell, there will be no life. So students, in today's lecture, we have done explanation of page number 4, 5 and 6. If you have any problem, you can ask me. Thank you so much. Allah Hafiz.